The overall goal of the fast nano-positioning system is to provide real-time structural information of biomolecules by combining single-molecule FRET experiments with rigorous statistical analysis.
This method answers key questions in the field of structural biology, such as the localization of flexible domains in macromolecules, where standard structural biology tools cannot be applied.
The major improvement of Fast-NPS compared to other FRET-based structural tools is that different dye models can be compared, yielding not only the most likely positions, but a three-dimensional probability distribution.
The main advantage of this technique is that it combines structural data from the protein database with quantitative single-molecule fluorescence measurements.
To begin the procedure, assemble a flow chamber and sample holder. To prepare the inlet and outlet screws, thread silicone tubing into hollow tab screws and cut the tubing straight on either end with a razor blade. Screw the tab screws to the sample holder. Align the quartz slide holes of the flow chamber with the sample holder threads. Gently tighten the glass holder screws to fix the flow chamber in place. Thread 20 cm long pieces of silicone tubing into the inlet and outlet screws. Wash the flow chamber with 500 microliters of PBS. Then, flush the flow chamber with 100 microliters of Nutravidin solution in PBS, ensuring that a droplet forms at the end of